அதுக்குள்ள போடனா அந்த ஏண்டா போடுற நைட்டி தள்ளுடா பேக்ல எடு ஹோம்வொர்க் எடுத்து
Good evening, students. Today we are going to discuss about autoimmune diseases. Autoimmune diseases. What is the meaning of autoimmune? Auto means a self. Auto means a self. Immune means you know very well. Immunity related to immune system. So some self antigens. I mean auto antigens. So some antigens also present in our body. That antigens, due to that action of that antigens, some diseases will be caused, which is called as autoimmune diseases. So autoimmune diseases are group of disorders which the tissues injury is caused by humoral or a cell-mediated immune responsible to self antigens. However, the autoimmunity so which means protection against self, but normally autoimmunity produces a tissue injury and tissue damaging in one's own body. Hence that autoimmunity is a misnomer. Autoimmunity is similar to hypersensitivity. What is the meaning of hypersensitivity? Next topic we are going to discuss it. So hypersensitivity means a allergic one. So autoimmunity also similar to the allergic reactions. In hypersensitivity, body react outside antigen, but in the case of autoimmunity, body reacts inside antigens. So antigens present in one's own cells are called autoantigens, which is also called as a self antigens. So just now I told outside antigens means some other bio things or microorganisms or other some dust or extra other particles, which is called as autoantigens, which causes some unwanted unwanted cases, which is called as outside antigens. Self antigens means that antigens are present in our body. That antigens make some diseases. That antigens are also called as self antigens. Clear? Autoimmunity, humoral or cell-mediated immune response against the self-antigens. So simply autoimmunity, we can say, autoimmunity means simply we can say humoral or cell-mediated immune response against the self-antigens. The both immunity will be involving in the autoimmune diseases. When the antigens remain in the scheduled place in our body, the antigen is not exposed to the immune system during fetal life. Hence, the system does not develop tolerance to the antigens. In later life, when this antigens leaks into the blood stream, the immune system recognizes the antigens as a non-self, which elicits an autoimmune response. So that autoimmune immunity was first discovered by the Mechnikov in Kinia. That question may be asked in Neet question. Kindly note it that autoimmune immunity was first observed by the Mechnikov. Mechnikov is a scientist in Kinia. The autoimmune disease was first observed by the Donath and Landsteiner. So kindly make the difference. Kindly try to understand the difference between tumor. Autoimmunity was first observed by the Mechnikov in Kineafix. Autoimmunity was observed by the Mechnikov, but autoimmune disease was observed by the Donath and Landsteiner. Clear? So what are some basic characters are autoimmune diseases means the autoimmune disease caused by the immune response against the self antigens. So that autoimmune disease caused by the immune response against the self antigens. So self antigens. The immune response are caused by the autoantigens is called as autoimmunity. The immune response caused by the autoantigens, which is called autoimmunity. That autoimmunity is classified into the two types. What are they? One is the humoral immunity, another one is a cell-mediated immunity. So humoral immunity, autoantibodies are produced. In humoral immunity, autoantibodies are produced against the autoantigens. For example, hemolytic autoimmune diseases, localized autoimmune diseases, systemic autoimmune diseases. So based on the immune response, antibody mediated autoimmune diseases. Autoimmune diseases are classified into the so many types. So based on the immune response, based on the antibody protection, or etc. etc. So before entering the types of autoimmune diseases, we should know some important characters of autoimmune diseases. Antigens, the immune system remains the silent for self antigens. This is called tolerance. The autoantibodies, in the case of humoral autoimmunity, the B cells are stimulated and autoantibodies are produced by the self-antigens. So that autoantibodies brings the tissue damage in two ways. What are they? One is the antibody-mediated autoimmunity and autoimmune complex-mediated immunity. Antibodies brings the tissue damage in two ways. One is the antibody mediated immunity, autoimmune complex mediated immunity. The antibodies directly brings about tissue damage. the autoantibodies directly brings about tissue damage is called antibody mediated immunity. Here there is no interaction of any other things. That antibody when enters, when stimulated, not enter, when it is produced by the immune system, at that time antibody directly involved the tissue damage, which is called antibody mediated immunity. For example, Myasthenia gravis, pernicious anemia. Those are the two examples of antibody mediated immunity. That autoantibodies combined with the autoantigens and forms the autoimmune complex. Autoantibodies combined with the autoantigens to form a autoimmune complexes. That autoimmune complexes cause the tissue damage. Clear? So that produced autoantibodies are combined with the autoantigens. Both will be present in our body only. That when the autoantibodies combined with the autoantibodies forms a autoimmune complex. Autoimmune complex. That autoimmune complexes cause the tissue damage, which is called autoimmune complex mediated immunity. So kindly make it clear, ma. So autoantibody mediated means that antibody alone directly involving for the tissue damage. That autoimmune complex mediated immunity means that both antibodies combined with the antigens, which makes a tissue damage, which is called as a autoimmune complex mediated immunity. Clear? For example for the autoimmune complex mediated immunity, lupus erythematosus, rheumatoid arthritis. Those are the some important examples. Some autoimmune diseases affect the tissues, particular organ or antibody. Some autoimmune diseases affects the tissues or a specific organ or the whole body. Accordingly, the autoimmune diseases are classified into the three types based on the affection. One is a hemolytic diseases, organ-specific diseases, systematic diseases. For example, hemolytic diseases means hemo means a blood related. Hemo means a blood related. So, the hemolytic diseases, the lysis of blood cells such as RBC, leukocytes, platelets, and etc. etc. Clear? So, RBC, leukocytes, platelets. So, it will be lysed, damaged by the auto antigens. For example, leukocytopenia, thrombocytopenia, pernicious anemia. Those are the example of hemolytic diseases. Here in autoimmune diseases, that examples and the name of the disease is very important. Kindly consider that one. So any examples may ask in Neet exams. So hemolytic diseases example is pernicious anemia, thrombocytopenia, leukocytopenia. And the next one is the organ-specific diseases affect only the particular organ. Organ-specific diseases. Name itself it having the answer. For example, thyrotoxosis, Addison's disease. Those are the few examples of organ-specific diseases. The third one is a systematic disease affect many organs. Systemic disease. Systemic disease means lupus erythematosus and rheumatoid arthritis. The two is the most important Neet question. Systemic diseases affect many organs. Lupus erythematosus and rheumatoid arthritis. For example, rheumatoid arthritis means mostly it affects the knee pain. It makes a knee pain. Affect a knee. Lupus erythematosus means it may be affect any organs like kidney or any other things. Systematic lupus erythematosus, SLE. Those also example for the systemic diseases. The autoimmune diseases are diagnosed by the many methods like examination of autoantigens, examination of autoantibodies, because both are involving for a tissue damage. So examination of autoantigens, examination of autoantibodies, X-ray imaging, CD or MRI scan. So those are the diagnostic method to identify the autoimmune diseases, and how which is treated that autoimmune diseases means a drug, surgery, replacement, transplantation, transfusion, and etc. But although we are treating like a drug, surgery, replacement, etc., we cannot say completely we cannot cure that autoimmune diseases. It may affect or may not affect. We can control that autoimmune diseases. The replacement or transplantation means when the organ is affected by the SLE, for example, kidney may be affected. So at that time, what we do? We have to transplant the kidney. So after that, this no problem, sir. You may ask the question. So after that, it may cause the same thing. So what we do? We have to maintain that controlled environment. For example, that kidney transplanted patient to avoid the salty things. So like that. So we can control that one. So treatment is a drug, surgery, retransplantation, transfusion, or etc. What are the causes of autoimmune diseases? Means sequenced antigens, lens antigens, sperm antigens, etc. So sperm lens antigens means a lens antigen is an antigen of islands. Sperm antigens means a spermatozoa developed during the puberty at the age of 13 to 15. The sperm antigens cannot induce tolerance during the fetal life. Hence, the sperm antigens enters the blood. It is not considered as a self. Hence, the self autoantibodies are produced. This occurs during the mumps infection. So many antigens can cause the autoimmune diseases. Lens antigens, sperm antigens, neoantigens. Neoantigens means modified antigens. Neoantigens, NEO. Neoantigens means modified antigens. Modified means that antigens are modified by the physical agents. For example, irradiation or a chemical agents, maybe a drugs or microbial agents. Clear. So due to that, any one of the thing, antigens may be altered. The altered antigens are no longer considered by the immune system as self-antigens. Hence, the altered antigens provoke an autoimmune response. The next, cross-reactive autoantigens. Cross-reactive antigens also cause the autoimmune diseases. Cross-reactive antigen means a foreign antigens which is structurally resemblance to the body antigens. Keep it in your mind. Cross-reactive antigen is a foreign antigen. You may ask one question here. So you only said, sir, self-antigen only make a autoimmune diseases. How that foreign antigens can cause autoimmune diseases means that foreign antigens, the structurally similar to the self-antigens. I mean the body antigens. As a result, the body antigens will react with the antibody produced. I mean autoantibodies will be produced. So automatically it affects the any organs like a human brain and a sheep brain. So antigen of human brain and sheep brain are a cross-reactive antigens. So those are the antigens causes the autoimmune diseases. So autoimmune diseases are classified into the 3 types. Just now I told. Okay, once again I reminding. So hemolytic, localized, and systematic. Other things, autoimmune diseases are classified based on the type of immune response. One is the antibody mediated, and one is the immune complex mediated, and last one is the cell mediated. Clear? So what are the other types of common autoimmune disorders? Means thrombophilia, rheumatoid arthritis, lupus erythematosus, Graves disease. Those are the common autoimmune diseases. The next one is transplantation immunology. That's all about the autoimmune diseases. Here, autoimmune diseases, that the disease's name is important one. The disease comes under which type of autoimmune diseases. You have to consider that one. After that, which one makes the autoimmune diseases? That thing you have to concentrate. So you have to concentrate what are the causative agent for the autoimmune diseases apart from the self antigens, and what are the types of autoimmune diseases based on the tissues or organs are based on the immune system. So what are the types of autoimmune diseases with the examples, and what are the common autoimmune diseases? You must study this one. That level is enough. The next one is transplantation immunology. Transplantation. What's the meaning of transplantation? Transplantation. Trans means a transfer. Trans means a transfer. So we are going to transfer the organs. We are going to transfer the organs. So study about that is called as a transplantation. Keep it in your mind, autoimmune diseases, transplantation, cancer, which is also present in your NEET syllabus. So kindly consider that one. So immunology is already I told it is like a max. If you understand, it is very easy. If you are not understand, too difficult to that one. But immunology is very, very important for to crack a NEET. Not only that to crack a NEET, it is if you know about that immunology means we can lead our life also. Because the biology, if you study that biology means it is used to get a high mark in NEET. Similarly, that biology will be support to lead the day-to-day life. Support to lead the day-to-day life. For example, in immunological chapter, what is the use of the transplantation? What is how that immunity will be developed? So if you know about that one means we can develop our immunity. Clear. So in this way, we can avoid the diseases. So you should know, you have to study more things related to immunology, not only to crack the NEET, to gain our knowledges. Clear. So transplantation immunology. So transplantation immunology, but now I am teaching based on your NEET syllabus only. So transplantation immunology means a study of response of immune system when a graft is implanted on an animal. So simply we can say implantation of tissue from one individual to another one. Implantation. So we are going to transfer, for example, kidney is, renal is failure. Failure means at that time what we do? So we have to transplant the another renal, another kidney from one person to next person. That is a solution for that one. Clear. So that organ is transplanted from one to another places. Otherwise, tissues will be transplanted from one to thighs. Tissues will be transplanted from one to another person. That is called as a transplantation immunology. Clear. So here, which tissue going to transplant, which is called as a graft, G-R-E-F-T. So the implanted, going to implanted tissue is called as a graft or a transplant. So the animal from the graft is taken means which is called as a donor. So donor means donating something. So where you are going to get the graft, which is called as a donor. Clear. So where you are going to place the graft, which is called as a, which one is called as a recipient or a host. So host or recipient means receiving the graft. Clear. With receiving the organs or tissues, which is called as a recipient. Donor means where you are going to collect the graft or organs or tissues, which is called as a donor one. The cells or tissues or organs are transplanted. So the transplantation, we can transplant organs or cells or maybe tissues will be transferred. For example, skin, what are the transplantation available means skin transplantation, kidney transplantation, heart transplantation, like that. Kidney we can transplant, heart, eye, and there's so many lung transplantations. So a lot of kind of transplantations are available. So next one is the types of transplantation. This is present in your syllabus. Types of transplantation. I mean types of a graft. Types means autograft, which is also called as a autogenic graft. Second one is a syngraft. Syngraft is also called as a syngenic graft. Syngraft is also called as a syngenic graft. And third one is a allograft, which is also called as a allogenic graft. Third one is a allograft, which is also called as a allogenic graft. Fourth one is a genograft, which is also called as a genogenetic graft. Genograft or a genogenetic graft. So that graft are four types. What are they? Autograft, syngraft, allograft, xenograft. We'll discuss one by one. The first one is a autograft. Autograft means a graft placed within the same individual. Auto means a self. Auto means a self. Auto means a self. Graft means the organ to be transplanted. So autograft means a tissue of individuals removed from one place from the particular individuals and which is implanted into the another place of a same individuals. Clear? So the particular tissue or organ, particular tissue is removed from the one place of a same person. That removed organ is placed in the same persons of another place, which is called as a autograft. This is called as a autograft. For example, plastic surgery or orthopedic surgery. Plastic surgery is an example for the autograft. That antigens present in the autograft, is the same that present in the body. Hence, that immune system recognize the autograft antigen is a self-antigens. Here, there is no immune response is elicited. That is important. Here, there is no immune response is elicited because that is a self-antigen only. Because that the tissues are transplanted from one to another place of same individuals. Clear? So as a result, autograft survives throughout the life. If there is an infection or a blockage or a circulation, it may prevent the survival of the graft. So without infection, without any other disruption of the graft, the transplanted graft will be survived throughout the life because new foreign antigen, if anyone is transplanted from one to another person, means at that time rejection maybe occurs immediately or maybe laterally. But the self antigen, self tissues will be transplanted from one to another place of the same individuals. Here, that same antigens is transferred from one to another place of same individuals. Here, there is no rejection. Clear? Because the self antigen only present in the tissues of transplant. The second one is a syngraft or a synergenic graft. Syngraft or a syngenic graft means graft takes place between the two genetically identified individuals. Syngenetic, same genetic. Syngenetic means the same genetic. So genetically identified individual of the same species. For example, the transplantation of organs or tissues between the identical twins or inbreed strains of the same species. Formerly, that syngraft also called as a isograft. Iso means the same. Iso means the same. That syngraft or a syngenic graft, which is also called as a isograft. So in isograft, histocompatibility antigens are identical. Hence, that the graft survives. It is not rejected. Here also, there is no rejection. In listen, autograft and syngraft, I mean isograft, there is no rejection because identical twins means having the same antigens. So while you are transplanting the organs or tissues from one person of identical to the another person of identical one means here there is no rejection occurs. So that graft transplanted organs or tissues can able to survive throughout the life because of no rejection. Graft survives and is not rejected because transplantation occurs between the identical twins. Clear. The third type is allograft, which is also called as a allogenic graft. Allo or allogenetic. What's the meaning of allo? Allo means same. I mean transfer of tissues between the two genetically distinct number of same species. The species is same, but genus is a different one. So here the transplantation is occur. It is called as a allograft. The allograft is formally named as a homograft because that transplantation occurs in the same species. Homo means the same. So the transplantation occurs between the same species, but different genus. Allograft histocompatibility antigens are dissimilar. Here we cannot say because the genus is a various one. Clear. Genetically different, but the species only same. Clear. So here antigens should show some differences. So histocompatibility antigens are dissimilar. Hence the immune response is elicited and the graft may be rejected. So the graft rejection occurs in the allograft and xenograft. Clear? Because of a dissimilar in histocompatibility antigens are present in the allograft. Clear? The last one is a xenograft or a xenogenetic graft. Xenograft or a xenogenic graft. The xenograft is a graft transfer of tissues between the two individuals or two different species. Clear? Two different species. The allograft means the same species. For example, in human being means one tissue is transferred from the one human being to the another one human beings. The same species, but genetically different. Genetically different means, for example, that is gene is transferred from me to the another one boy. That boy is not related to me. Clear. So the species is the human species, that is the same one, but family wise it is a different one. That is a genetically variant. It shows us some genetical variants, but species is a same one. Here that transformation is occurs. That is called as the allograft. Clear. Xenograft means that species transplantation occurs between the two different species. For example, animal to human being. The transplantation maybe occurs from the animal to human being. That is called as a xenograft or a xenogenic graft. Transplantation of tissues between the two individuals or two different species. So that's why the xenograft is also called as a heterograft because the two different species is involved. So heterograft. Here that the histocompatibility complex of antigens are so different because the species is entirely different. So the histocompatibility complex antigens are so different antigens. The graft is more vigorously rejected than the allograft. Clear? Than the allograft. So kindly keep it in mind. There is no rejection in autograft and xenograft. Important question. There is no rejection occurs in autograft and xenograft because that histocompatibility antigen is a identical one, same antigens. So there is no rejection. In autograft, self antigens. Xenograft, identical antigens are identical one. But in the case of allograft, histocompatibility antigens shows a dissimilar. Because for example, kidney is transplanted from the one person to another person. So which rejection maybe occurs within an hour or within a day or after 10 years. So that chance is high for a rejection due to the dissimilar antigens. Similarly, in xenograft, In xenograft, its histocompatibility complex antigen shows a so difference in the antigens. So that rejection is vigorously in xenografts because the transplantation occurs the unrelated animals. For example, human big heart may be transplanted to the pig heart may be transplanted to the human one, like that. Unrelated transplantations. Clear. That's all about the types of antigens. So here important terminology, graft acceptance means when the graft tissues remains alive. So which is called as a accepted process is called as a graft acceptance. When the transplantation is made between the genetically identical individuals, the graft survives and lives as healthy, because that graft is accepted by the body. Graft rejection means when the transplantation of tissues, the transplanted tissues are dies, the graft is said to be rejected. The process is called as a graft rejection. So when the transplantation is made between the genetically distinct individuals, the graft tissues dies and decays. The graft is rejected because the antigens of graft and host are different, and the immune response is elicited, leading the death of the graft tissues. Clear? This is called as a graft rejection. The graft rejection is occurs in the xeno and the allograft. Acceptance. In all things, acceptance will be occur, but rejection is high chance for rejection is allograft and xenograft. I think that level is enough for you for Neet. The next one is a tumor one. So tumor means yesterday we discussed about a lot of diseases. Lot of diseases. Here, what is the infection diseases, non-infection diseases, or etc. etc. After that, we discussed about the HIV. HIV means can cause the disease named as AIDS. After that, we discussed about the autoimmune disorders. So likewise, tumor means a continuous growth of cells, uncontrolled continuous growth of cells. That is called as a tumor. In your syllabus, which is this topic is given in as a cancer. So tumor is an independent, autonomous, uncontrolled growth of tissues containing the mass of abnormal cells, which is called tumor, which is also called as a cancer. In your syllabus, they gave as a cancer. That cancer and tumor are both are the same. The tumor means the independent, autonomous, uncontrolled growth of tissues. So it produces a mass of abnormal cells. That producer group of cells are abnormal one. It is a group of cells. That uncontrolled growth of abnormal cells are collectively called as a cancer, which is also known as a tumor. Although the global level 7 million deaths per year is due to the cancer only, because a lot of cancer causing agent we are intaking, either food or maybe air or maybe any other thing. For example, regularly in day-to-day life we are using so many things. For example, vehicles, industries, and so many things are releasing many gases, smokes like that. Unwanted gases also may cause cancer. Similarly, that CFC, chlorofluorocarbon, which is present in the refrigerator, most probably we are using that refrigerator, cooler, or etc. etc. So that refrigerator releases a CFC, chlorofluorocarbons. So while we are using that one, automatically it may cause the cancer. So apart from that one, the oil, repeated oil we are using to fry the some other things. While you are eating that one, automatically it is also causative agent of a cancer. Like that, so many causative agents will be there. Clear. Pesticides, okay, we are using that chemical pesticides, or etc. etc. So while we are taking that chemical pesticides or chemical fertilizer used to food items, while we are taking at that time, automatically which is also can cause the cancers. So lot of it. So early stage of tumors does not exist any symptoms. The tumor is recognized in later stages only. That is a problem. That is a problem. That's why the death is occurs at a high rate. But early detection also possible. But we have to more consciously. We have to more consciously observe that one. At that time only we can easy to observe that one. We can easily to identify the early stage cancer. If it is identified in early stage, it is possible to eliminate the tumor. What are the types of tumor means? Based on the spreading, the tumors are classified into the two types. One is a malignant tumor, another one is a benign tumor or a non-malignant tumor. The first one, what is the meaning of the malignant tumor means? That tumors are rapidly spread from the one place to another place. It gives a pain. Malignant tumors are rapidly spreads easily. Also gives a pains, which is called as a malignant tumor. The types is important one, kindly note it. Another one is a benign tumor or a non-malignant tumor, which grows slowly and does not spread from one place to another place. So that non-malignant is opposite to the malignant. It is not spread from one place to another place. It grows too slowly. Further, tumors are classified many types based on the nature of tissues affected. Nature of tissues affected. The common type is a malignant and non-malignant. For example, that another common types of cancers are carcinoma. Carcinoma means the tumors arising from the epithelial cells. Epithelial cells means the skin cells, outside cells. That is called as a carcinoma. Sarcoma. Sarcoma means SARCOMA. Sarcoma means the tumor arising from the mesenchymal tissues, means a connective disease, and endothelial cells. The tumors arising from the connective and endothelial cells, which is called as a sarcoma. The next, lymphoma. Lymphoma means a tumor arising from the lymphoid tissues. Tumor arising from the lymphoid tissues. It is called as a lymphoma. The last one is a leukemia. Leukemia means tumor of white blood cells. WBC. The tumors occurs in the white blood cells, which is called as a leukemia. So those are the important NEET question, ma. So kindly note it that malignant. What is the meaning of malignant tumor, and what's the meaning of a non-malignant tumor? Sarcoma means occurs in the epithelial cells. That is important. Sarcoma means occurs in the connective or endothelial cells. Lymphoma means occurs in the lymphoid tissues. The leukemia is also important question. Tumors occurs in the white blood cells called as a leukemia. Questions may ask like this: the tumors appears in leukemia. The tumor appears anywhere in the body. It may appear in the skin, lungs, gastrointestinal tracts, breast, uterus, brain, blood, etc., 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 etc. It may cause at anywhere. What are the basic properties of tumor? Means it is uncontrolled growth. It grows and multiply rapidly. Uncontrolled growth. That's why it will forms swelling like appearance. So initial stage it won't give a pains. Laterally it gives a pain. In mitosis, the cell will be controlled, undergoes the cell division. But here uncontrolled growth of cells. because that mitotic division will be affected in cancer causing patients. So there is no control of mitosis. What will happen? Mitosis means, you know very well, continuous cell division, but it should be a controlled environment. There is no control of mitotic division means that cells will be uncontrolled growth and forms a mass of group of cells. So it shows a swelling like appearance, which is called as a cancers. When it matures, at that time only it gives a pains. The tumor cells behaves like a embryonic cells. They originated from the undifferentiated cells by the dedifferentiation. The tumor cells carry out none of the functional or normal adult cells. Tumor cells lost their contact inhibition. Tumors avert the immune system. Surely it will affect the immune system. The immune cells are not very efficient in detecting tumor. Those are some important properties. What are the causes of tumors? Next, causative agents for tumors. Causative agents means spontaneously developed. Spontaneous means natural. So certain normal cells are spontaneously transferred from one to another tumor cells. During the transformation, they undergo a dedifferentiation and assumes the embryonic cells. Then some viruses cause the tumors. Certain viruses transform normal cells into the cancerous cells. The certain viruses transform normal cells into cancerous cells. Those viruses are called as oncogenic viruses. Oncogenic viruses. That oncogenic viruses produce tumor-specific antigens on the surface of transformed cells. So what are the virus can cause the tumor means adenovirus. That name of the virus, kindly note it. Adenovirus can cause the tumor, tumors. That adenovirus can produce the tumor-specific antigens on the transformed cell surface, which is responsible to make tumors. Then chemicals, many chemicals, methylcholine, it can cause the variety of tumor antigens. Many chemicals comes under the carcinogen, like mercury chloride and so many dyes, dyeing agents, which is also comes under the chemicals. The next one is the oncogenes. Oncogenes means oncogenes are set of genes inducing tumors. And the oncogenes are found in viruses as well as in the mammalian cells. The oncogenes are present in the both mammalians and the viruses. That oncogenes are present in viruses is called as a viral oncogenes. If that oncogenes are present in the mammalian cells, that is called as a cellular oncogenes. That is also important, that cellular oncogenes means that oncogenes are present in the mammals called cellular oncogenes. If the oncogenes are present in the virus, which is called as a viral oncogenes. Next important is the tumor antigens. What's the meaning of tumor antigens? The antigens developed by the tumor cells, which is called tumor antigens. Important thing, kindly note it, that antigens developed by the tumor cells, which is called tumor antigens. The next, fetal antigens. Fetal antigens means the differentiated cells undergoes a dedifferentiation to become tumor cells. Results of the dedifferentiation, the adult cell become the embryonic cells. Adult cell consists of embryonic cells. That fetal antigens, I mean that carcinoembryonic antigens, CEA, carcinoembryonic antigens in cancer of intestine. And the tumor specific antigens means TSA, tumor specific antigens, which is a unique tumor cells. It can reject the tumor crops. So they also called as the tumor specific transplantation antigens. Clear? So how the tumor is caused means that a particular protein is important for the tumor causing, that namely P53, protein 53. That is an important NEET question mark. Protein 53, P shortly called as the P for protein, P53. So that P53 proteins are present in the all mammalians, in humans. In all humans having the P53 proteins normally. When it is activated, at that time only it becomes a cancer cells. At that time only it becomes a cancer cells. So P53 proteins are a carcinogen 1 when it is activated. So present in everywhere in normal human beings in silently. When which is activated through the physical radiations, X-ray, UV rays, or etc., or maybe chemicals. While we are taking the chemicals, the carcinogenic chemicals, at that time P53 will be stimulated. So that P53 once it is stimulated means it causes a cancer cells. It produces the cancer cells. Clear? So P53 is the important one. So how we can treat that? What are the factors involved in that tumor immunity means what are the factors involved in the tumor immunity means the tumor cells formed in our body. It is delete by the following factors: plasma cells, B cells, D helper cells, macrophages, killer cells, antibodies, lymphokinases. Those are the some factors involved in the tumor immunity. Immunotherapy also we can give a cancer. That is a treatment of cancer. So tumor is prevented and treated by the so many ways. For example, injection of BCG means a corynebacterium. Injection of a BCG vaccine or a corynebacterium pervum is a beneficial effect on tumor. For example, intrathoracic injection of BGC has a significant effect on lung cancer, like a vaccine. It will be act as a vaccine. And administration of LAK cells and IL-2 gels. And interferon therapy. Interferon therapy means interferons reduces the certain types of cancers like renal cancer, myeloma, sarcoma, lymphus, and hairy cell leukemia. So that things will be cured with the help of interferon therapy. How it will be cured means what is the mechanism involves means interferons function as a primarily as a anti-proliferative agent. Anti-proliferative agent. Proliferation means a multiplication. So when that interference is induces involved that anti-proliferation, the proliferation is prevented. The next one is a vaccination. So certain types of cancers will be controlled by the vaccination. The vaccination. For example, that virus is induced tumor cells will be produce the antigens. So which virus can induce the cancer cells? Cancer, that viral antigens are collected, which is deposited on the surface of the tumor cells. The small dose of the same type of tumor causing virus is injected into the tumor patients. That antibodies will be produced. That antibodies are react with the tumor antigens and the tumor cells are destroyed. So vaccination, BCG vaccine, interferon therapy, those are the important treatments. Diagnosis means how we can diagnose the tumor means carcinoembryonic proteins. Identification of carcinoembryonic proteins, oncofetal increases in immune complexes. Those things, if you identified, means surely we can diagnose as the cancer ones. Clear? So that's all about the cancers. The next one is a hypersensitivity reaction. Hypersensitivity reaction. So what's the meaning of hypersensitivity reaction? Hypersensitivity. So hypersensitivity means simply we can say allergy. Hypersensitivity, simply we can say allergy. The violent reaction of immune systems leading to the severe symptoms, even death. Clear ma? Violent reaction of immune system leads to severe symptoms and even death in a sensitized animal when it is re-exposed to the same antigens for the second time. So hypersensitivity means a allergy. So that severe reactions will be occurs due to the some antigens, some factors involving or causing the hypersensitivity reaction. If the re-exposed of the same antigens are the same factors, at that time that allergy is formed. What are the factors causing the allergy? Means a hypersensitivity. That factors are called as allergens. So allergens, that is the important question. Allergens means a hypersensitivity or a allergy causing causative agents, which is called as a allergens. In clinical terms, a hypersensitivity is called as a allergy. The famous example is a penicillin injection. Penicillin injection. The hypersensitivity is caused by the penicillin injection. Sometimes in hospitals, patients dies immediately after penicillin injection. This is due to the allergy. That allergy is caused by the drug is called drug allergy. So allergy will be caused by the so many things. For example, some people's while we are taking some people's brinjal will be act as a allergens. In some people that non-veg items like chicken act as a allergens. For example, in some people's while they are taking the chicken means automatically that body skins are becomes a reddish in color. That is called as a chicken allergy. For example, in some people, if any drug is taking, if any kind of drug is taking, immediately that swelling, itching, and skin becomes a redness. So those are the symptoms. So that person is called as that person's having the drug allergy. Some people, the castor oil allergy. Castor, which makes a allergy. A brinjal also makes a allergic reaction for some people. So allergic, so that brinjal called as allergens. The drug is called as allergen because makes a allergy. That it will be depends upon the person to person. So penicillin injection is an example for the drug allergy. The immune system devised by the nature primarily to protect the human body against the pathogen. Sometimes the systems becomes over enthusiastic, which brings the discomfort. That phenomena is similar to the over enthusiastic severant who tried to kill the fly sitting on the king's nose with a strong sharp knife. So our over enthusiastic reaction of the immune systems makes the allergic reactions like a hypersensitivity reactions. So because the immune system always directed towards the protection of host one, but hypersensitivity immune response becomes injurious to the host one. So normally immune system's immune response are a protection of a host one, but in the hypersensitivity reactions, immune response becomes the injurious to the host one. Hence that immune response, you see, immune response becomes a destructive process in a hypersensitivity. In normal protective immune response, that antigens, bacterium, virus all will be killed or maybe neutralized. But in the case of hypersensitivity, the cells of host cells will be killed, or host itself is damaged. Host itself, it is a damaged. Hypersensitivity is a changed reactivity of a immune system. So what are the factors cause the hypersensitivity? Means just now I told the hypersensitivity causative agents are called as allergens. It may be extrinsic factor. I mean, introduced to the body from the outside. Otherwise, intrinsic factors. Intrinsic in means inside. Factors within the body. That is called as an intrinsic factor. So hypersensitivity caused by the extrinsic factor or intrinsic factors. What are the factors cause allergic reaction? Means drugs such as penicillin, aspirin, sulfonamide, and etc. And the airborne particles also cause the allergy. For example, pollen grains, house dust mite. For example, some women are while cleaning the day, that particular persons may affect the some cold or etc. That is called as a dust allergy. And sports and animal donates the scale and the feathers of the animals. And foods of fuels, for example, a fish, strawberries, brinjal, or etc. That is also the brinjal is some cause some allergic. Some people cannot take the brinjal due to the allergic reactions. And the infectious organisms like a bacteria, virus, fungus. For example, some people cause some allergic reaction by the fungus. And blood transfusion. Blood transfusion means a mismatched blood groups causes allergy. That's why we have to transplant the same blood groups. What are the symptoms of hypersensitivity? Means rashish. So mostly rashish and redness of the skins. Skin become a redness, rashish, and itching problem. Surely it will cause. For example, brinjal cause allergic reaction means itches and swelling of a particular place. For example, who are having the drug allergy at that time, if you are going for hospital to take a treatment for the particular diseases, means what they do, immediately the nurse will inject the small drop or small amount of particular medicine will be injected, and they will make a circle around that injected part. After half an hour, they will check. After half an hour, they will observe your result. Any itching, they will ask you if any itching or any redness or any swelling is appeared or not. They will check it. After that only that particular injection will be injected to you. Clear? Because the drug is also allergic reaction. So like that, we have to check it. So itching, swelling, redness, rashes, atopy, diarrhea, and vomiting. Those are also symptoms of a allergic one. Swollen lymph nodes. Those are the important symptoms of allergic reactions. And what are the common hypersensitivity reaction types? What are the common types? Means hypersensitivity produces the variety of reactions. For example, the reaction may be manifest immediately. Some reactions. For example, if you are taking any other unwanted thing, I mean allergens, immediately it shows us some reactions. For example, drug, otherwise drug allergy, it shows immediate reaction within half an hour. Clear? within a half an hour. For example, that reactions may manifest immediately within a minutes or within a minutes or in a delayed state after 24 to 48 hours. The reaction is caused hypersensitivity, may be local reaction or a systemic reactions. When the symptoms appears in a restricted area or areas, that reaction is called a local one. So when the allergic reactions occurs in the restricted area of human body, which is called as a, symptoms occurs in the restricted area of human body, which is called as a local reactions. When the symptoms are affect all organs of systems of the body, that reaction is called as a systemic. It will be affect the all parts of body, which is called as a systemic hypersensitivity reactions. That reactions are so many types. For example, anaphylaxis, transfusion reactions, erythrobetolysis, Arthur's reactions, serum sickness, Mantis reaction, context, dermatitis, Graves diseases. So mostly the types of hypersensitivity is in the two way. One is the based on the time taken for the reaction. Another is based on the different mechanism of pathogens. So mostly that hypersensitivity reactions are classified into the two types. One is the based on the time taken for the reaction. Another one is the based on the different mechanism of pathogens. The based on the time taken reactions, that hypersensitivity reactions are categorized into the two types. What are the means? One is the immediate hypersensitivity, another one is the delayed hypersensitivity. Sorry. Immediate hypersensitivity means then the immune response or immune reactions manifest in a short duration in minutes. The hypersensitivity is called as the immediate type. That one allergen, when the allergen is entered into our body, at that time it shows the immediate allergic reaction, which is called as the immediate hypersensitivity. Name itself is having the answer. For example, drug allergy, just now I told penicillin belongs to that category. Hay fever, HOA. Hay fever is another example of a immediate hypersensitivity. So that immune response appears as well as disappears rapidly. So appear rapidly, similarly disappear rapidly. So inflammatory response occurs in the few minutes. Inflammatory responses. That the immediate hypersensitivity involves the interaction of antigen and antibody. How we can prevent the immediate hypersensitivity means which is inhibited by the antihistamine drugs except for Arthur's rejection. That immediate hypersensitivity reaction is inhibited by the antihistamine drugs. In this way, we can reduce the action of immediate hypersensitivity reactions. The next one is a delayed hypersensitivity. Delayed hypersensitivity means the name itself is giving the answer. Dilated. Later. So when the immune response or reactions manifest slowly after 24 hours to the 72 hours, that kind of hypersensitivity is called as a delayed hypersensitivity types. Familiar example of delayed hypersensitivity is a Montorux reaction. Means clinics for a drug-nosing primary complex in the children's. That reactions mostly appears after 24 to after 1 or 2 days, after 24 to 48 hours, mostly within 1 or 2 days. So while we are taking that allergen when the allergen mass entered into our body, at that time it shows some allergic reaction after 1 or 2 days, which is called as a delayed hypersensitivity reactions. That reactions are characterized by the erythema and induration. That immune response are very slowly and lots longer. It produces erythema, induration, and lymphocytes infiltration. That delayed hypersensitivity involves the reaction between the antigens and T cells. But the immediate hypersensitivity involves the antigen and antibody. Here the reactions occurs between the antigen and T cells. So that delayed hypersensitivity handled by the T cells. Hence, the delayed hypersensitivity is called as a cell-mediated immunity. That is an important question. Delayed hypersensitivity mediated by the cell-mediated immunity. That delayed hypersensitivity is suppressed by the corticosteroids. Suppressed by the corticosteroids. Those are the important types of hypersensitivity reactions. So that that is based on the time taken, and next that is a classification of hypersensitivity based on the mechanism of pathogens. Mechanism of pathogens, that hypersensitivity reactions will be classified into the 5 types. First one is a type 1, type 2, type 3, type 4, and type 5. Type 1 is the anaphylactic hypersensitivity reaction. Foreign substances causes the anaphylactic hypersensitivity reactions. The type 2 is antibody dependent cytotoxic hypersensitivity reactions. Type 3 is immune complexes mediated hypersensitivity reactions. Type 4 is cell mediated hypersensitivity reactions. Type 5 is stimulatory hypersensitivity reactions. Clear? Those are the 5 types of hypersensitivity reactions. Just you know the name itself, that is enough. Because in our syllabus, they have that majorly 2 types of dilated and immediate hypersensitivity reactions only. Although you should know the different mechanisms, different types of hypersensitivity reactions based on the mechanism of pathogens. Type 1, anaphylactic. Type 2, antibody dependent cytotoxic hypersensitivity. Type 3, immune complex mediated hypersensitivity. Type 4, cell mediated hypersensitivity. Type 5, stimulatory hypersensitivity. At least you should know the name itself. This is called as a hypersensitivity reactions. The next one is the vaccines. The vaccines and the vaccinations and the vaccination scheduled already we discussed it. Because already we discussed it. Vaccines are which is prepared by the vaccines are antigenic preparation of organisms. That vaccines are prepared by the organisms, either heat killed or inactivated, or that antigenic properties inactivated. So vaccines are antigenic preparation of organisms, bacteria, viruses. We can prepare any kind of organisms like a bacteria or any other things, which is used to prevent the infectious diseases. That is called as the vaccines. For example, polio vaccine is prepared from the inactivated polio virus. Like similarly, that corona vaccines are prepared from the corona killed coronaviruses, or maybe that organism product. For example, spike protein also, which is used to produce the coronaviral vaccine. Clear? So that either vaccine means organisms or a product of organisms which is used to produce the vaccine, which is used to prevent the or treatment of infectious diseases, which is called as a vaccine. Like here, that vaccine preparation, either live vaccine or killed vaccine, inactivated organisms will be used for the preparation of vaccines. That vaccines we can give it in the oral form or maybe injection form. The types of vaccine preparation also already we discussed. The common vaccines are polio vaccine, BCG vaccine, DPT vaccines, which is used to improve the immunity and etc. With the vaccines are used to prevent the diseases or which is used to cure the infectious diseases. That homologous vaccines, heterologous vaccines, a subcellular vaccine, inactivated vaccines, etc., etc. Those are the some important types of vaccines. And the vaccination schedule also, that diphtheria vaccine, DPT vaccines, that things already we discussed in the previous class. I think 2 days before class already we discussed. Vaccination schedule also, I explained with the help of that vaccination schedule, because that vaccination schedule is prepared by the government agencies or physicians. Clear? For example, from the birth itself, we have to give the vaccines from the day 3 and up to 12 days. We can that vaccine we can take vaccines to prevent the so many diseases. Clear? That is called as a vaccination schedules. Already that thing also we discussed. So that's all about the some important diseases. So next class we will discuss about the alcohol and addiction of alcohol or etc. etc. I think tomorrow we'll complete the immunology chapters. Immunology chapter, which is also known as applied biology chapters, which is also called as a human health and diseases chapters. Clear? So thank you, students. Tomorrow we will discuss about the alcoholism or alcohol addiction. Thank you. Today class is over. Thank you very much.